I work in the Department of Chemistry under Dr. Matthew Lauper, and my research is using plant-derived compounds to combat antibiotic resistance. So over the past couple decades, we've had an increase in antibiotic resistance, and the roots of those causes come from misuse of antibiotics in healthcare as well as in agriculture. So in healthcare, that might be a doctor who misprescribes or overprescribes or prescribes an antibiotic for a viral infection that's instead of a bacterial infection. And in agriculture, that would be farmers who pump antibiotics into the feed for their livestock, that when we then consume the meat, we're secondhand exposing ourselves to those antibiotics. Another component is patient compliance. So if you're prescribed an antibiotic for 7 days, but you feel better after 3 and you stop taking it, that can cause selective pressure for genes that support the resistance to antibiotics. The end results are pretty staggering. C. diff, which has been treatable through antibiotics since the mid to late 1900s, the death rates from that are up 400%. 1 out of every resistant case that's reported is fatal, and there are over 200,000 cases reported every year. And that's the number from 2013, and it's only increased since then. 1 out of every 5 hospital room visits that are drug-related are from antibiotic-resistant cases. And when you consider the opioid epidemic that we're in, the fact that 1 in 5 drug-related visits are still due to antibiotic resistance, that's a pretty impressive number. So the genes that are being selected for cause overexpression of efflux pumps. And these efflux pumps have been shown to be the most popular mechanism that bacteria cells use to be resistant to antibiotics. So we're looking at 2 bacteria cells here. The darker ring on the outside would be the membrane of the bacteria. The green portion inside the cell, blue obviously the outside. The cylindrical shapes on the membrane of the cell represent efflux pumps. Efflux pumps work like inverse vacuums. They're used in normal cells to pump out toxins, get rid of anything that the cell doesn't want or need anymore. So a normal cell has a few because they need them, and a cell that overexpresses can have 2 to 8 times more of those. So when antibiotics are introduced into a normal cell, the cell has the capability of pumping out some of the antibiotics, but not enough. A lethal dosage is still left, and the cell will die. When the antibiotics are introduced into a cell that overexpresses for these efflux pumps, the antibiotics will get completely pumped out, and the cell will be able to survive, thus passing on its resistant genome to more bacteria cells, and that's essentially how this resistance happens. So the pump that we look at in particular, there's lots of kinds, but we look at the ACR-AB Toll-C pump that's found in E. coli cells, as well as a few other gram-negative bacteria. The ACR-AB Toll-C pump is composed of 3 different proteins. The upper protein faces the outside of the membrane, Toll-C is its name, and if you were to look down the middle of it, it looks like a paper towel tube, so it's just a long, narrow channel that is non-discriminate and will allow anything to pass through it that comes its way. The component of the ACR-AB Toll-C pump that faces the inside of the cell is called ACRB. It uses a proton motive force, which is essentially using a change in, or a difference in charge across that inner membrane to power the pump. So that's the pump mechanism of the whole protein structure of the transporter. And so it'll use that proton motive force to power the compounds to go in it, pump it to Toll-C, and out of the cell. ACRA is the 3rd component, and that's just an accessory protein that binds the 2 together. And it stays inside the periplasm or between the 2 membranes, inner and outer membranes. So the basis of our research is based on the fact a few different things about plants. 1, plant cells have been around much longer than animal cells. Plants can't go to the doctor. They can't get antibiotics. They have to come up with their own ways to combat infections. Plant cells, had to adapt, and since we eat produce all the time, or we should be, there's a lower risk of toxicity to the compounds that we may find that work to combat antibiotic resistance. And there's also a convenience factor. It's easier to find these things in grocery stores as opposed to having to make them in some sort of laboratory and make a synthesis of some sort of compound that way. Up to 80% of the world's populations to this day still rely on plant medicines. We've used them in the past. We get morphine from the opium plant, and we get, morphine from willow tree bark and some other drugs that we use. And so it's not uncommon for us to look to plants for their medicinal properties. Last summer, Dr. Lauper's lab started this research, and they took about 30 different plants, shoots, fruits, leaves, and seeds, tongue twister, And they took them through a process that I'll explain, and they tested them for hits. The only positive hit they found of those 30 was green tea. So when I came into the lab that fall, knowing that green tea is chock full of catechins, polyphenols, theaflavins, I chose produce that also had concentrations of those things, one or a few of them or all of them. And that's where I got my list that you can see on either side of the screen. And it's as simple as banana and as weird as that crazy tea that I still can't pronounce. But what we do is we take them into our lab, we process them by drying them, and then we use a sock slit extractor, which works basically like a coffee machine. And that's what's being shown up here. So we take our produce that's been dried, we put it into a paper thimble, we insert it into our sock slit extractor, and then we use hot methanol to steep out all the compounds that are in the produce. That then drains down into a round bottom flask, and we use that to dry the methanol away, get all that gone because we don't want it. And it just gives us a really rich composition of the produce. So if we were to apply these extracts into a cell and look at the cell, we wouldn't be able to see whether or not it's able to cause inhibition of that efflux pump. There's just not a way for us to be able to do that. So instead we have to use a model. We use the ethydium bromide assay to test whether or not the cell is able to efflux with the presence of our extracts. So we take E. coli cells, and then we apply our ethydium bromide, which when given the chance to bind to DNA will cause fluorescence. And we put them into a reader that can detect that fluorescence or not. So in a cell where there's no efflux occurring, the extract has successfully inhibited those pumps from working. The ethydium bromide will accumulate inside the cells, bind to the DNA, and will give us a fluorescence indication. In cells where efflux is occurring, those pumps are not being inhibited. There's nothing wrong with it. The extract isn't working. The ethydium bromide doesn't get the chance to accumulate, and we'll see no fluorescence. So of all the extracts I tested, I had one positive hit, which was with yerba mate. So on the y-axis here, we have relative fluorescence units, so that's our fluorescence that I was referring to earlier. And on the bottom, we have time. So we watch these cells over 30 minutes to see how much growth occurs in that, or how much fluorescence can occur in that time. The uppermost line is heat-killed cells. Those are cells that have been killed, and they don't have any energy or any way to run the pump, to create energy to run that pump, and so they will retain all the ethydium bromide that comes their way. The next line, the gray line, is CPZ, which is another positive control. CPZ disrupts the proton motive force on the inside of the membrane that powers the pump to work. So again, the pump's unable to pump out anything, and we see accumulation of ethydium bromide over time. The bottommost line is our water, which is a negative control. So those are healthy, happy cells. They pump out whatever they want to pump out, and they pump out that ethydium bromide, no binding occurs. The middle line, the green line, is my yerba mate. It went from about 600 to 1,300 relative fluorescence units, which even though it never quite reached the fluorescence of our positive controls, it never plateaued. And so we found that interesting enough to pursue and look into. But what is yerba mate? Yerba mate comes from a plant called Ilex paraguariensis. It's grown in Central and South America, and it's very common down there. They drink it like we might drink water. Really, they have it everywhere. It comes from a plant that looks about like that. Well, it does look like that. And they take the leaves and they dry the leaves. They really don't process them much. There's no oxidation, there's no fermentation, they just dry them. You can buy it in the store. This is the most common brand, and it's a brand that we use in our lab. And you steep it just like tea. It comes in a little tea bag. The reason it became popular is because it has a lot of caffeine in it, and then it also has some really great microbial effects to help with keeping your body and the toxins in your body at a good, manageable level. Unlike Camellia sinensis, which is the plant that we get green tea, oolong tea, black tea, and white tea from, there are absolutely no catechins in yerba mate. When the people in our lab have continued to look at green tea, they've been pretty interested in catechins and the effectiveness of the catechins on creating inhibition. And knowing that yerba mate doesn't have any catechins in it, we know that we're probably looking at some other sort of compound, which is pretty interesting. So yerba mate, the extract of it, is this thick, green, goopy, it's just full of dozens of compounds. And so we don't really know which compound is causing the inhibition. So what we want to do is separate out our extract. And to do that, we use high-performance liquid chromatography. So we take a solvent gradient. We use a methanol water solvent gradient where we go from about 0% methanol and about 100% water to the vice versa. So by the end, we're pumping about 100% methanol through there. We inject our sample, which is, ooey gooey. It's got stuff all together. You can't tell what's what. It pumps through a column which uses the difference in polarity of those compounds to separate it out. And it will go through a detector which will detect the absorbance of those compounds. And then we ask the machine to fractionate it for us. So it splits up the sample into 95 different test tubes over 4 hours. And then based on that, we can run ethidium bromide assays like we have before and test which fraction is causing the inhibition. So this was the chromatogram that I got from the HPLC that I ran on my yerba mate. I want to keep your eye focused on the blue line. We're looking at absorbance values on the y-axis and time from 0 to 240 minutes. So as the sample ran, it spit out different compounds, and you can see, when a large absorbance occurred, a high absorbance rate occurred on the bigger spikes. So that's when different compounds came through. The lower levels were probably lower amounts of those compounds coming through. Then if I can direct your eye to the green bars, those are the results of the ethidium bromide assays that we ran on the different fractions. So the same assay that we ran before, where we apply ethidium bromide to a well with cells and all that, instead of using our different extracts, we used the different fractions. So we ran 95 of those. And the ones with the highest bars, there's 3 main ones and then one kind of in the middle, and the rest are pretty low. The 3 high ones are the 3 that we're the most interested in. Those cause the most accumulation of antibiotics, and so we're pretty sure that there's a compound in one of those fractions that's causing the inhibition of the efflux pumps. So moving forward, we know that these fractions can cause accumulation of ethidium bromide, but that's not very clinically relevant. We need to know that they cause accumulation of antibiotics in order for us to really be interested in these compounds. So what we're going to do is use a liquid broth dilution assay, which basically you just use carbonicillin which is our antibiotic of choice, as opposed to ethidium bromide, and test it similarly. So these 5 wells will be filled with carbonicillin at different concentrations. The one that's the darkest would represent no carbonicillin, so no concentration of it, and those cells are allowed to grow over time. The well will look really cloudy. The one that's the most white, the carbonicillin's at the highest concentration, so it'll be able to hopefully kill those cells, and we'll see much less cloudiness. Then over here, these 3, we'll be comparing mainly against the middle one, the 8 micrograms per milliliter concentration of carbonicillin. These wells will have the cells, the carb, and then they'll also have our fractions. So if in these wells, they are less cloudy than the regular 8 microgram per milliliter control, if they're less cloudy, then we know that the presence of the fraction enhances the effectiveness of the antibiotic, and those fractions, those compounds, the compound in those fractions are clinically relevant. Moving forward, also, we're going to be using when we first used our extract, we got a hot methanol extraction from it. We're also going to be using a cold methanol extraction, and we're going to be comparing the HPLCs of those 2 and see if we can cross out any compounds, because the cold methanol did not create a hit the first time through. So we know that anything that came out in the cold methanol won't come out, and also came out in the hot methanol, that's not an active compound. So we're going to be able to cross a few compounds off and hopefully put our finger on exactly which one is causing the inhibition. I want to thank the University Dayton Honors Program, Claire Booth Luce, Dr. Lauper, and the Lobsters. It's been a really fun summer with all of them, and I can't wait to continue this research for the next year or so. So thank you. I'll take any questions. Yeah.
What kind of cells are you doing for the last experiments that you just mentioned?
E. coli cells.
Okay.
Yeah, we're using the same line of E. coli cells, AG100, for all of our experiments.
Okay.
Yeah.
Why did you choose the antibiotic you chose?
Carbonicillin. I think we've been using carbonicillin in all the antibiotic tests that we ran. Like whenever we try to test any we've done a few different types. We've done some well diffusions, some liquid broth dilutions, and I think it's just the amount of literature that we have on it. We know the lethality of it with E. coli, so I think it's more well known to our lab. We have literature to back up the concentrations that we're using.
Is there a high level of resistance to that one?
I don't know. I'm actually unsure. I know that a lot of beta-lactams, is that right? I don't want to say anything I don't actually know. But I know that there are a few families of antibiotics that have seen a larger amount of resistance. I'm not sure if carbapenicillin falls into that antibiotic or not.
Thank you.
Yep.
Is the efflux pump, is there evidence that that is antibiotic specific, or is it fairly generalized?
It's a very general pump. It'll pump out anything. The reason we chose to look at ACRIB Tulsi as opposed to other pumps don't span both membranes. They're not as non-discriminant, and it takes a lot more, like they might pump it into the periplasm, that space between the two cell membranes, but not all the way out. Whereas ACRIB Tulsi spans both. So we know that it's one of the bigger of the pumps. We also don't really know if it's using the pump in particular, so that's another prong of research that we might have to go down, is whether or not it's using ACRIB to cause inhibition, or if it's going through some other mechanism that's allowing accumulation. If it's going through another efflux pump or some other sort of mechanism.
Are there blockers for that pump?
Well,
Synthesized pharmacological blockers that would be specific to the pump?
I'm not sure. We are looking at using aptamers to block Tulsi to see if we can kind of like cork in the bottle and see if we can block anything from being able to get out of that. That's another prong of research that we're looking down too. That CPZ is able to disrupt the proton motive force, and I'm sure there are other ways to do that as well. Yeah.
It's just a comment. I was at Whole Foods the other day and I saw the yerba mate,
Yerba
and I
mate.
thought of you.
You'd be surprised how often I get that. Yeah, Lindsay.
So going down that route, how has your research then affected your personal life? Do you
Listen, I am the big coffee drinker, but no, I really like yerba mate. It tastes it's like a really nice taste, and it has so many positive benefits. It's kind of one of those things, if you can choose coffee or tea, even though it has the same amount of caffeine, but it releases over longer periods of time, whereas caffeine and coffee gives you the big spike. Yerba mate is more just a steady go along. So yes, I do drink yerba mate. I try to as much as I drink coffee.